The overall aim of this procedure is to obtain pure astrocyte cultures by isolating and culturing mixed cortical cells from P1 to P4 mouse pups. This is accomplished by first harvesting brain cortices of mouse pups and removing the meninges. The second step is to cut the cortex into small pieces, dissociate the cortex pieces by trypsinization and trituration to obtain single cells, and to plate the single cells on poly-D-lysine-coated tissue culture flasks. Next, after 7 to 8 days, astrocytes, microglia and oligodendrocytes will have formed different layers, and the separation of astrocytes from microglia and oligodendrocytes is achieved by shaking the tissue culture flask on an orbital shaker. The final step is the trypsinization and harvest of the remaining astrocytes, which are then replated in a tissue culture flask. Ultimately, 12 to 14 days after the first cell split, astrocytes are plated at the appropriate concentration for experiments, and cell purity can be examined by immunocytochemistry using astrocyte-specific markers.
The method outlined here is based on the astrocyte culture preparation from rodent neonatal brains originally described by McCarthy and DeVellis in 1980.
This method can help answer key questions in the neuroscience field, such as questions regarding the interplay between astrocytes and neurons, as well as the role of astrocytes in ZNS disease, where astrocytes become activated and contribute to inhibitory scar formation.